In this lecture, I'm going to talk about diuretics. So let's get right into it. So diuretics, also known as water pills, simply promote the elimination of water from the body. Now, in order to understand how diuretics work, first we need to review the basic physiology of nephron. So as you may already know, nephron is the structural and functional unit found within the kidneys. Each nephron has 5 distinct regions that have specific function. First, we have Bowman's capsule, surrounding a network of capillaries called glomerulus. Here blood comes in through the afferent arteriole, where components of the blood get filtered out of the capillaries into the capsule. Filtered blood is then carried away from the glomerulus by efferent arteriole. Next, glomerular filtrate, which contains mostly water, amino acids, glucose, sodium bicarbonate, and electrolytes, flows through proximal convoluted tubule. In this region, almost 100% of the amino acids, glucose, and about 90% of bicarbonate get reabsorbed, as well as about 65% of electrolytes such as sodium and potassium, along with water. As a side note here, remember, wherever sodium ions go, chloride and water tends to follow. The next major region through which the filtrate travels is the descending and ascending loop of Henle, which passes into the medulla of the kidney. Now, the descending limb has walls permeable to water, but impermeable to sodium. So as the filtrate travels down, water leaves, but sodium tends to stay, making the filtrate more concentrated. On the other hand, the opposite takes place in the ascending limb, particularly in the thick limb, which is permeable to sodium but impermeable to water. So as the filtrate travels up, about 25% of sodium gets reabsorbed passively, as well as via active reabsorption mediated by the sodium-potassium 2-chloride cotransporter. Next, the filtrate travels through the distal convoluted tubule. Here, about 5 to 10% of sodium chloride gets reabsorbed, mainly via sodium chloride cotransporter. This part is also relatively impermeable to water, so at this point, the filtrate becomes more diluted. Finally, the filtrate travels through the late distal tubule and collecting duct. Here we find two types of cells, the principal cells and the intercalated cells. Now the functions of the principal cells are regulated by two hormones, aldosterone, which increases sodium reabsorption and increases potassium secretion, and antidiuretic hormone, which increases water permeability and thus increases water reabsorption. Now, on the other hand, the intercalated cells are regulated by aldosterone, but their primary function is to secrete hydrogen ions and reabsorb potassium. So now let's switch gears and let's talk about diuretics. Diuretics simply increase the volume of produced urine by acting on different parts of nephron. So in other words, diuretics help us to remove salt and water from the body, which makes them very useful for treatment of hypertension and abnormal fluid retention referred to as edema. Now, based on their mechanism of action, diuretics can be divided into 5 major classes, and these are carbonic anhydrase inhibitors, loop diuretics, thiazide diuretics, potassium-sparing diuretics, and osmotic diuretics. So let's look at each class in more detail, starting with carbonic anhydrase inhibitors. Carbonic anhydrase inhibitors produce their diuretic effect mainly by reducing reabsorption of bicarbonate in the proximal convoluted tubule. Now let's zoom in on the epithelial cell of the proximal tubule to see how they exactly do that. So to the left of the epithelial cell we have lumen, where urine is being formed, and to the right we have interstitium, which contains blood vessels. Now here bicarbonate reabsorption is initiated by the action of sodium-hydrogen ion exchanger, which allows sodium to enter the cell in exchange for hydrogen ion. Next, reabsorbed sodium is pumped by sodium-potassium ATPase out of the cell, while the secreted hydrogen ion combines with the luminol bicarbonate ion to form carbonic acid. Carbonic acid is then rapidly dehydrated to carbon dioxide and water by an enzyme carbonic anhydrase. Now, carbon dioxide enters the epithelial cell by simple diffusion and gets rehydrated back to carbonic acid by intracellular carbonic anhydrase. Finally, intracellular carbonic acid dissociates to form hydrogen ion, which can be transported by sodium-hydrogen exchanger to the lumen, and bicarbonate ion, which is transported out of the cell. Now, when carbonic anhydrase inhibitor comes around, it inhibits carbonic anhydrase enzyme, and as a result, bicarbonate gets retained in the lumen. Since bicarbonate reabsorption utilizes the sodium-hydrogen exchanger, inhibition of carbonic anhydrase leads to significant reduction in proximal tubule sodium reabsorption, which ultimately results in mild diuresis. Now, the reason why the diuretic effect is only mild is because the majority of the sodium that doesn't get reabsorbed in proximal tubule still gets reabsorbed by distal parts of the nephron. However, this late reabsorption also leads to increased potassium secretion, which is one of the side effects associated with this class. Additionally, because bicarbonate is a major component of the acid-base buffering system in the circulation, its wasting leads to increase in plasma acidity known as metabolic acidosis. Now, one of the most popular drugs that belongs to this class is acetazolamide. However, due to its relatively weak diuretic effect, acetazolamide is commonly used for its other pharmacologic actions. Now, let's move on to loop diuretics. So, loop diuretics, just like their name suggests, work in the loop of Henle, specifically in the ascending limb, where they inhibit sodium-potassium 2-chloride cotransporter. Out of all diuretics, these agents produce the greatest diuretic effect because as much as 25% of sodium gets reabsorbed in the ascending limb, and at this point, other parts of nephron can no longer compensate for increased levels of tubular sodium. So now, let's zoom in on the cell that lines the thick ascending limb to see what exactly happens there. So upon reaching the thick ascending limb, some of the filtered sodium, potassium, and chloride get reabsorbed by sodium-potassium 2-chloride cotransporter. This, among other things, leads to accumulation of potassium within the cell. However, potassium also tends to leak out through potassium channel back into the lumen, which contributes to a more positive charge there. Now, this positivity in the lumen produces electrical driving force for paracellular reabsorption of cations such as magnesium and calcium. So, when loop diuretic comes around and blocks sodium-potassium 2-chloride cotransporter, we not only lose sodium and water, but also potassium, calcium, and magnesium. Now keep in mind that potassium loss takes place mainly in the late distal tubule, where sodium-rich fluid enhances sodium-potassium exchange, and I'll talk about this more later. Drugs that belongs to this class include bumetanide, ethacrynic acid, furosemide, and torsemide. Now when it comes to side effects, besides causing electrolytes imbalance, loop diuretics can cause rapid and excessive reduction in blood volume, also known as acute hypovolemia. This can lead to hypotension, shock, and even cardiac arrhythmias. Next, because loop diuretics inhibit sodium-potassium 2-chloride cotransporter, which also happen to exist in the inner ear, their use has been associated with the damage to the hearing, also referred to as ototoxicity. Lastly, ethacrynic acid and furosemide in particular compete with the transport of uric acid at the same site, thus blocking its secretion, which ultimately can lead to hyperuricemia, and thus worsening of gout symptoms. Now let's move on to thiazide diuretics. So thiazides are probably the most commonly prescribed diuretics. They work mainly in the early part of the distal tubule where they increase sodium chloride excretion by inhibiting sodium chloride cotransporter. However, since over 90% of sodium is reabsorbed before reaching the distal tubule, in general, thiazides produce rather weak diuresis. On the other hand, thiazides are capable of causing vasodilation, which reduces peripheral vascular resistance by a mechanism that unfortunately is not clearly understood yet. Now, let's zoom in on the epithelial cell of the early distal tubule to see what exactly happens there. So here, sodium and chloride enter the cell via sodium chloride cotransporter. Once inside the cell, sodium gets transported to the blood via the sodium-potassium ATPase, while chloride diffuses out on the other side via chloride channel. Now, when thiazide diuretic comes around and inhibits this sodium chloride cotransporter, the result is decreased sodium chloride reabsorption, and ultimately, since water follows the salt, we get increased urine output. Drugs that belong to this class include chlorothiazide and hydrochlorothiazide, which are structurally similar sulfonamide derivatives, and we also have chlorthalidone, metolazone, and indapamide, which are often referred to as thiazide-like drugs because they don't have true thiazide ring but share the same mechanism of action. Now, when it comes to side effects, just like loop diuretics, thiazides can cause hypokalemia, which results from increased delivery of sodium to the distal parts of the tubule. Moreover, similarly to loop diuretics, thiazides can also lead to hyperuricemia by interfering with uric acid transport, which in turn may exacerbate gout. Now, while loop diuretics can cause calcium excretion, leading to hypocalcemia, thiazides, on the other hand, can cause the opposite, that is hypercalcemia. There are two mechanisms that are thought to be responsible for this. First takes place in the proximal tubule, where thiazide-induced volume depletion leads to a compensatory increase in sodium reabsorption, which in turn produces electrical gradient, which leads to passive calcium reabsorption. The second mechanism involves the sodium-calcium exchanger located on the basolateral side of the distal tubule. So because thiazides block sodium chloride cotransporter, there will be decreased concentration of sodium inside the cell, which then in turn causes this sodium-calcium exchanger to bring in more sodium in exchange for calcium, which is then excreted into the bloodstream. Lastly, thiazides may worsen glucose control, leading to hyperglycemia, and they may also elevate cholesterol, leading to hyperlipidemia. Some of the mechanisms thought to be responsible for these effects include decreased insulin secretion and insulin sensitivity, as well as increased hepatic glucose production. Now let's move on to potassium-sparing diuretics. So potassium-sparing diuretics work primarily in the collecting tubule, where they inhibit sodium reabsorption and potassium excretion. Although these potassium-sparing agents are relatively weak diuretics, they are often used in combination with diuretics from the other classes in order to enhance their effects. Now let's zoom in on the collecting tubule principal cell to see what exactly happens there. So principal cell has separate channels for sodium and potassium, as well as sodium-potassium ATPase on the basolateral side. Under normal conditions here, sodium enters the cell through sodium channel and then is transported by sodium-potassium ATPase into the bloodstream in exchange for potassium. Now, because this sodium entry predominates here, the lumen negative electrical potential is generated and chloride is driven into the bloodstream through paracellular pathway. This in turn drives potassium out of the cell through potassium channel. So as a side note here, this illustration should also help you understand how loop diuretics and thiazides lead to potassium loss. So increased sodium load caused by loops and thiazides enhances sodium reabsorption, which makes lumen more negative. This in turn generates driving force for increased potassium secretion and ultimately lower potassium levels in the bloodstream, hence hypokalemia. Now going back to potassium-sparing diuretics, agents within this class can be separated into two groups based on their distinct mechanisms of action. So the first group works simply by blocking sodium channel, which results in decreased sodium-potassium exchange and thus retention of potassium. Drugs that belong to this group include amiloride and triamterene. Now, the second group, on the other hand, works by antagonizing aldosterone. Aldosterone is a hormone which gets into the cell, binds to an intracellular receptor, and stimulates transcription of genes encoding sodium channel and sodium-potassium ATPase, thus increasing their expression. In other words, aldosterone leads to increased reabsorption of sodium and water and increased secretion of potassium. So, because aldosterone antagonists compete for binding to that intracellular receptor, the result is reduced synthesis of proteins that activate sodium channels and decreased number of sodium-potassium ATPases, which ultimately leads to potassium retention. Drugs that belong to this group include spironolactone and eplerenone. Now, when it comes to side effects, as you may already guess, hyperkalemia is the biggest problem, especially when these diuretics are combined with other drugs that can also increase potassium levels. Lastly, due to its chemical structure that resembles our natural steroid hormones, spironolactone can stimulate receptors for testosterone and progesterone, leading to effects such as menstrual irregularities in females and gynecomastia in males. Now, before we end, I wanted to briefly discuss the last class of diuretics, namely osmotic diuretics, which work by interfering directly with osmosis. As you may recall from your basic biology class, water has tendency to move across membrane from lower osmolarity, or the dilute side, to a higher osmolarity, or the concentrated side. So these osmotic agents are filtered from glomerulus. They are very water-soluble, or hydrophilic, and they undergo very limited reabsorption due to their large molecular size. This leads to increased osmolarity of the tubular fluid and thus decreased water reabsorption. Now, because osmotic diuretics increase water excretion rather than sodium excretion, they are not very effective for treating edema caused by sodium retention. Instead, they are mainly used for reduction of intracranial pressure, promotion of urinary excretion of toxic substances, as well as promotion of urine production in patients with acute kidney failure. Examples of osmotic diuretics are mannitol and urea. As for the side effects, use of osmotic diuretics can lead to significant fluid changes such as volume overload or dehydration and thus electrolyte imbalances. And with that, I wanted to thank you for watching. I hope you enjoyed this lecture, and as always, stay tuned for more.